Dear students, this is Kuldeep Singh Gaur from Department of Zoology, Government Meera Girls College, Udaipur. Today I am going to speak on the introduction of cell in terms of cytoskeleton, nucleus, the chromosome structure, centrioles, etc. Now, what is this cytoskeleton? The cell has to maintain a specific shape, size, and structure. This is enabled by presence of some proteinaceous and filamental structures, which forms a network inside the cytoplasm, which helps the cell maintain its shape. The next one is what are cilia and flagella? The cilia and flagella, they are the outgrowth of the cell membrane, and they are in fact primarily they are motility organelles, but cilia do perform many functions apart from the locomotion. Cilia works like or causing movement of either the cell or the fluid surrounding it, or it moves the mucus inside our body when they beat in a coordinated fashions. The flagella, they are comparatively longer than cilia. If we cut the CS of cilia or flagella, we come across this type of structure. The outermost is plasma membrane, and we have 9 peripheral doublets made up of microtubules. These doublets, they are made up of 13 protofilaments, which are made up of alpha and beta tubulin, which I am going to tell you later when we'll deal this organelle in detail. So, there are 2 central microtubules which are present in the center, and the 9 doublets, they are connected to the core area by radial spokes. The doublets themselves get connected by interdoublet bridges. Then there is a difference between prokaryotic or bacterial flagella or the flagella of eukaryotes, in the sense that bacterial flagella are made up of flagellin and protein silene. The core area of the cross section, or the cilia or flagella, is called as exonyma. It possesses number of microtubules running parallel to its axis. The exonyme contains 9 pairs of doublets which are radially arranged peripheral microtubules. 1 pair is centrally located. That's why the microtubular arrangement in cilia and flagella is 9 plus 2. Then the central tubules are connected by bridges and enclosed by sheath. The central sheath is connected to one of the tubules of each peripheral doublet by 9 radial spokes, as I've told you earlier. Then peripheral doublets are also interconnected by linkers. Both cilium and flagellum emerge from centriole-like structure called as basal bodies, which are present immediately beneath the plasma membrane. They are the mother of cilia and flagella, but their microtubular arrangement is 9 plus 0. However, the microtubule arrangement of cilia and flagella is 9 plus 2. This is the central difference between basal bodies and cilia and flagella. Very similar to basal bodies are centrosome or centrioles. Centriole is an organelle which occurs in doublet and they are cylindrical in structure. Both the centrioles and centrosome lie perpendicular to each other like this. Each has a cartwheel microtubular organization, and we'll be dealing it in detail when we take up this organelle separately. Made up of 9 peripheral triplet fibers. Each fiber is a triplet. Adjacent triplets are linked, and the central part is proteinaceous and is called as hub. There isn't any microtubule present in the hub. That's why the microtubule arrangement is 9 plus 0. The hub is connected with tubules of peripheral triplets by radial spokes made up of proteins. The centrioles are the prime reasons for forming basal bodies, which gives rise to cilia and flagella. The centriole, they are responsible for formation of spindle fibers and esters during cell division in animals. However, they are absent in plants. Come to nucleus, was discovered long back in 1831 by Robert Brown. It contains chromatin and is stained by basic dyes, and the material which is stained by basic dyes is given the name chromatin by Fleming. The interphase nucleus, that is the stage when the cell is not dividing. At this time, nucleus has extended, unfolded, highly elaborate nucleoprotein fibers called as chromatin. Apart from chromatin, the nucleus has nuclear matrix, which is formed by nucleoplasm, and also they have typical nucleoli, which in number 1 or more than 1. The nucleus consists of outer nuclear envelope, which is double membrane bound, and the two membrane, they are not continuous. In fact, they meet at points called as nuclear pores. This is the rough endoplasmic reticulum. Many a times the ribosomes are also present on the outer nuclear membrane. Nuclear envelope consists of two parallel membranes, and they have a space in between of the range of 10 to 50 nanometer. This space is called as perinuclear space. Nucleus, that's why divide the cell into two compartments, one containing nucleoplasm and another containing cytoplasm. The outer nuclear membrane is continuous with the endoplasmic reticulum, which bears ribosomes on it, as I have drawn in the diagram. At a number of places, the outer and inner membrane fuses, and it fuses to form the nuclear pore, from where the nuclear pores are the passages through which movement of material in and out of the nucleus takes place, in both the directions. Normally, we have one nucleus, but many a cell and organisms they have more than one nucleus in their cells. Like for instance, in paramecium, we have one micronucleus and another is macronucleus. The micronucleus is for this is micronucleus. This is meant for reproduction in paramecium, whereas the macronucleus this is meant for metabolism. There are two types of cells uh, in terms of presence of multiple nucleus. One is called as syncytial cells. They are formed by fusion of cells and the plasma membrane they coalesces and multiple nuclear condition is created. Another is sinusoid. Here the nuclear division occurs, but it is not followed by cytoplasmic division. But there are certain cells which even do not have nucleus in them, and one example is mature erythrocytes of many mammals and seed tube cells of vascular plants. The nuclear matrix contains one special structure called as nucleolus, another is chromatin material. The content of nucleolus is continuous with the rest of the nucleoplasm. Nucleolus is not a membrane-bound structure. However, it is the site for formation of ribosomal RNA. That means the chromatin which is placed at the site of nucleolus contains rDNA. This rDNA later combines with proteins to form granules, and these granules are called as ribonucleoprotein particles. Larger and more numerous nucleoli are present in cells which are actively engaged in protein synthesis. This is the diagram of nucleus. You can see the outer nuclear membrane and the inner nuclear membrane. In between the outer and inner nuclear membrane is perinuclear space, and this scattered inconspicuous thread-like material is called as chromatin, and this is endoplasmic reticulum, which is laced with so many of the ribosomes. Now come to the chromosome. The indistinct chromatin material which is laid inside the nuclear membrane during interphase gets associated with DNA, histone proteins which are basic in nature, and also some non-histone proteins for stabilization purpose, along with some RNA, and condenses, that is dehydrates to form chromosomes. A single human cell has approximately 2-meter long DNA distributed among its 46 or 23 pairs of chromosomes. However, chromosomes are visible only in dividing cells. Look at this chromosome. This is metaphysic chromosome. At the center, these are the two chromatids joined by two centromeres. Each chromatid has centromere in between, and above the centromere, coin-like structure called as kinetochores are present. Based on the presence of or the position of the centromere, the chromosomes can be divided into metacentric chromosomes. When the chromosome contains the centromere exactly at the middle, it is metacentric chromosome. When the centromere is slightly displaced from the center, towards the distal end. Such a chromosome is called as submetacentric. This is submetacentric chromosome. Then comes acrocentric, little more displacement of uh, centromeres makes it as J-shaped during the anaphase. This is acrocentric. And when the centromere is present at the tip of the chromosome, it is called as telocentric. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location that gives appearance of a small fragment-like satellite like this. This is not constriction, primary constriction. This is called as satellite. This is secondary constriction. Apart from these organelles, the cells, both plants and animals, have many membrane-bound minute vesicles called as microbodies, which contains various enzymes. We'll study those enzymes during the detailed study of these organelles. Thank you so much.